The cell membrane separates the cell from its environment, just like our skin does for us. And it regulates what can go in and out of the cell. Cytoplasm is everything on the inside. That would be all of your guts and your blood and everything on the inside of your body is the cytoplasm. And then inside the cell, we have kind of the brain, which is the containing of the DNA, which is the nucleus. It contains all the genetic material. So these are just 3 definitions. We're not getting into any abstract concepts. The cell membrane is the skin of the cell. The cytoplasm is everything inside the cell. The nucleus contains the DNA. Everybody okay? Fantabulous. Here we had a picture. I'm not going to have you label anything on the test. So going into the cell membrane, the skin of the cell. Ooh, my heart is skipping beats. Give me just a second. All right, there we go. All right, so the cell membrane is the skin of the cell. It regulates what can come in and out. And it is the makeup of the cell membrane that kind of prevents some stuff from getting in and more importantly, prevents stuff from leaking out, like our skin. We don't want our organs just like leaking out of our bodies. That would be terrible. And just like our skin, this cell membrane is capable of repair to a degree. If it's injured too much, obviously the cell is going to die. Just like if your skin is injured too much, then you can't repair it. So it governs the movement of molecules. Now in chapter 4, we're going to discuss how molecules move back and forth through. For chapter 3, we just need to know that molecules move back and forth. The cell membrane, the skin of the cell, is made of this phospholipid. Lipid meaning what? Fat. We have hydrophilic and hydrophobic ends on these skin. Hydrophilic faces where? Outside. Because remember we compared this to our bison. Remember this is our little bison. In fact here, Rita can even give you a little visual here. Let's have some fun. Like put little horns. Little bison horns. I know those are not looking like bison horns, but you get the idea. Help visualize it. The bison heads love a fight. Hydrophilic love. Everybody who's afraid is where? Inside at the tail end. So the heads are hydrophilic. The tails are hydrophobic. Phobic meaning fearful or repelled. Now, we are water-based creatures. Right? Our body makeup is about 70% water. How well does fat and water mix? It does not. And so this is what really regulates what can come into the cell. Because if the majority of the things trying to get in are water-based, and our cell membrane is fat-based, that makes it pretty difficult. And so we have to have these special kind of doors and channels and hallways and secret passages for this stuff to get in past this fat membrane. We also have some other things that are kind of wedged in, like cholesterol. Now this is the good cholesterol. Cholesterol is a ginormous molecule, and we wedge it in for support. It's kind of like support beams for the cell membrane. We've also got things like sugars and amino acids. All these are helping to support the structure while maintaining the cell's ability to change its shape. That's it.
Everybody the ability to help change the shape.
Yeah, so some of these structures, we need the cell to have the ability to change shape, but we don't want it to, you know, like if I have a cell, and he's got to flatten himself out like this to get through and out of a blood vessel, I don't want him to collapse and just lose everything. So this whole cell membrane allows him to move out of the blood and then come back. So it's just there to help support the cell membrane and allow him to change shape without damaging it.
You said that's the cholesterol, right?
So that's everything: cholesterol, carbohydrates, the amino acids, all of the above. Did anybody watch the video, Mr. Anderson, cell membrane? He's actually the one I stole the bison thing from.
Yeah, he's one of those that talks about like there we go, boom, and the chemistry. So it's a PowerPoint. It's on your PowerPoint.
I think it's on the PowerPoint. We might watch it afterwards. So, because he does a really great job of kind of drawing it out. He's a far better artist than I am. So here we have a great picture of a cell membrane. It almost looks like marshmallows floating on the top of a hot cocoa, the heads, the hydrophilic heads. But we can see that there's stuff wedged in there. So here we have proteins, we have these sugars, these carbohydrates that help to maintain the structure, but we also have little secret passageways that allow things to move through. Here we have another protein structure. So it's really, really complex and kind of complicated, considering it's just a single cell. We had a couple of different proteins. Again, this is just terminology. I'm just looking for definitions here. Integral proteins. These proteins create little channels. Now, whenever we say the word channel, we're talking about some sort of opening, some kind of passageway, like almost like a canal.
Like
right?
a ship channel.
Like exactly like a ship channel. That's what Doug was trying to think of. Moving the ship from one area to another. That's what these channels do. They help to move molecules from either in to out or out to in, just past this membrane. The types of channels we have, selective. Now, what does the word selective mean to you?
It only like you select a certain, like you decide.
Only certain molecules.
Yeah, only certain. You get to pick and choose.
A few.
Only a few. Only very specific molecules are allowed to use this door. I think of it like the VIP, right? If you've ever gone to a concert or gone to anywhere and bought VIP tickets, I highly recommend it. It's amazing. And once you go VIP, you'll never go back to general admission ever, ever, ever again. Back pre-COVID, my significant other, huge comic book fan, huge, humongous. So obviously we would go to Comic Palooza every year. Anybody ever been? It's a lot of fun. It's a lot of fun. We dress up in costume. We do the whole 9 yards. The one year, we worked on our costumes for like 7 months prior to. VIP, baby. Pass the line. Heck yeah. That's a selective pour. You got to show them the ticket, and only certain people are allowed in. Only certain molecules can come in. Pours, on the other hand, you still need the ticket, but it's general admission. So it's not nearly as selective. Again, you can't go to Comic Palooza without a ticket, but you can't pass the line without the VIP. Selective pours are passing the line. Or selective channels, pours are general admission. Does that make sense to everybody?
Yes.
Comic Palooza is amazing, by the way. If it ever comes back, ever, I highly recommend it. It's a hoot. They do. We're not really into the anime. We just like the.
What about the Renaissance fairs?
We do the Renaissance, the Ren Fest. We haven't gone in a lot of years. We just prefer, because we like to go and meet the celebrities
Nice.
and get our photos. That's kind of our thing, is we collect photos with celebrities. So,
The Renaissance Festival.
oh my goodness, which ones have I not? I've met Stan Lee
Wow.
4 times.
That's awesome.
I got 2 photos and 7 signed prints from Saint Stan, which is fabulous. Absolutely. George Takei. Milo Ventimiglia. Anyone watch This Is Us? Huh?
He's so handsome.
No, not fibromyalgia. I did not meet fibromyalgia. Tim Curry.
Oh.
I got to meet Tim Curry.
I just know one person.
Yeah. Adam West, the original Batman. Met him. Trying to think. I have so many. So I'll think of some more. But that's kind of our groove.
You know, I would love to meet Johnny Depp.
I've not met Johnny Depp. I
met his mom.
did get to meet, although his real name right now is escaping me. Golly. He played Daryl Dixon on The Walking Dead.
Oh, Warren.
Damn. I know, my Doug is not on the wheel right now. No, it's not Reagan. I'll think of it. I gotta stop thinking about it and then I'll think about it.
Norman Reedus. That's what Melissa said.
Thank you, Zoomer. Norman Reedus.
There we go. I knew it was
What did I say? I said Roman.
Got my picture with him. So it's pretty great. It's pretty fun. Anyway, here's another picture showing the complexity of this cell membrane. Again, all of these proteins. We have also, remember, our peripheral proteins out to the side. They also help with structure. And really, all of the things that are embedded in this phospholipid bilayer will either do one of 3 things. Provide stability, allow the cell to change its shape, allow passageway for other molecules. Primarily, those are the 3 things that all of these structures that are embedded do.
Change shape, allow passage of what else?
Change shape, maintain structure, and allow passageway. And this is, I'm just telling you that all of the proteins, the carbohydrates, the cholesterols will do kind of one of 3 jobs. Here we have the glycocalyx. Glyco meaning what?
Sugar.
Sugar. So you really are sugar and spice and everything nice. Everybody, even you, even though you're not a little girl. Because you know that poem.
No, I didn't. I'm not a little girl anymore. That's fine. I don't want to be accepted.
Because you know the poem, girls are sugar and spice and everything nice. Little boys are, what was it? Something and puppy dog tails. Something and snails and puppy dog dirt and snails. Yeah. You really are made of sugar. We're all sweet like sugar. Every one of our cells has this special individual unique coding. And this is what allows other cells to identify it and recognize it and say, okay, you belong. You're supposed to be here, or you don't belong, stranger danger. It's the glycocalyx, this special, unique sugar coating that is unique to the cell and allows for recognition. It also helps cells to move over each other. helps cells kind of stick and then kind of roll over each other. Because we got a whole lot of cells and there is not a whole lot of room inside of us. So it gets a little tight in there and we've got to be able to move. That's our glycocalyx. We've got a couple of families in this particular category. We have our cell adhesion. These help cells move past each other, right? Adhesion means to stick. Cells kind of stick together and roll past each other. Like if I'm walking and there's another cell coming this way, I can kind of, and then we kind of roll over each other like this, and then he can continue that way and I can continue this way. But the other thing that this cell adhesion does is it's kind of the beacon to signal other cells. So if you get an area of inflammation and I need backup, this cell adhesion is kind of the CD, the radio that I can contact other cells and say, hey, I need help in this area. And then they'll all come and help. Membrane receptors, these also help with cells recognition. It's really, really important that my body be able to recognize who is supposed to be in here. Because when my body stops recognizing who's supposed to be here, he just starts attacking everyone. And then you got big problems. If your body starts attacking you, holy smokes, how do you stop that?
Is that like lupus?
Lupus is an example of a disease where your immune system starts attacking you. That's a problem, because if your immune system decides that you are a foreign invader and he just starts attacking you, think of it like you have the military. And the military is looking, because your immune system is like the military of your body. The military is constantly on the lookout for foreign invaders. But what happens if the military stops recognizing each other and thinks that they are the foreign invader? And they're just taking each other out. Suddenly, guess what? You don't have no more military. And then guess what? Foreign invaders are like, hey, look, nobody's guarding that border at all. Let's go in and have some fun. Terrible, terrible, terrible. And then we have cell movement. Show me the movement of cilia. That was our sea anemone, right? Think about the clownfish hiding in the sea anemone. Like Nemo. And it's crowd surfing stuff. And we have flagella. That's the tail that moves in kind of a whip-like forward motion. Mammals only have one particular cell that have a tail. Sperm. We got a lot of cells that have cilia. We got cells in our nose, we've got cells in our lungs, we've got cells in our trachea. Anytime we've got to be able to crowd surf stuff. We've got these cells, cilia in our ovaries that very gently move the egg into position. So cilia, flagella.
I have a question about that.
Yes.
On the test yourself, there was a question that was talking about which one is most common in mammalian, cilia or the flagella.
Well, if there's only one type of cell in half of the population of mammals that has a flagella, but every single one of us has cells that are ciliated in our lungs, in our noses, and our trachea, which one do you think is more common in the mammal? Bam.
Sorry, I guess I was
No,
so tired.
I'm just here to
Yeah.
help you critically think it through. That's what I'm here for. All right. What questions can I answer about any of that information before we move on to the next?
I don't know. It was on the one about
Oops.
Do you need to know anything about the
Okay, we haven't gone over that PowerPoint yet.
Okay.
That's in part 2.
Okay. Oh, it's that's the part 1.
Oh, yeah, that's where we start talking about, are you talking Calviole or Christe? Oh, that's right. I don't know why this particular PowerPoint is missing that.
Like one's the chemical, like one slide, the other one's this one, this one.
Oh, yeah, I couldn't find the.
Because the virus one,
That wasn't there.
Yeah, we also did. It was like the.
All right, so you have a question about chemical signaling. What?
Do we need to know about?
Well, you need to know like about the cell adhesion molecule form of the calyx, that sort of thing, just the definitions. Here I've got a cell. Here I have something that I need to move out. How do I do that? I have this ability that this can now come and form with the lipid membrane. And now my membrane kind of does this, and this can open up, and my little thing shoots out into space. That's what we're talking about there. Just shoots it out into space. Pew! Calviole. What?
Someone in the chat said, I like the way she said about.
It's the Canadian in me.
It's the accent.
You can take the girl out of Canada. Can't take Canada out of the girl.
Place we look better here.
Well, I've lived here longer than I lived there. But as far as like it better, they're apples and oranges. Two completely different states of mind. So they're a lot more progressive up there. That's all I'm going to say about that. All right, so now that we've discussed the cell membrane, the skin of the cell, now we're delving inside. That's what part 2 is, just all the stuff on the inside. Cytosol, jelly-like substance. Cytoskeleton is exactly what it sounds like. Cyto, the cell,
The skeleton.
skeleton, it's the skeleton of the cell. Organelles, tiny organs, and inclusions is just a void, just a big space. So the cytosol is this jelly-like fluid. Think of your cells kind of like a water balloon, and that's what allows them to have this kind of ability to shape and move and be kind of dynamic. It's not just a hard round ball.
So our cytosol and cytoplasm basically.
Yes. Cytoplasm is everything, like everything on the inside. The organs, the skeleton. When you say cytoplasm, you're just referring to
Like a generalization.
a general inside the cell. Cytosol is specific for the jelly-like substance.
Okay.
It's made of proteins. It helps our organelles just kind of float around and move around the cell. The cytoskeleton, exactly what it sounds like. You guys are doing skeleton in a medical terminology. Cells have a skeleton too. We've got creepy rule of 3, 3 parts to our cytoskeleton. Microtubules, intermediate fibers, microfilaments. Papa bear, mama bear, baby bear.
Was there anything highlighted on this slide? It's not on your testable material.
This slide, I think what I did was I took these three individuals when they have their individual slides and I highlighted the stuff you needed to know on those slides.
Oh, okay.
Of these three, which is the largest cytoskeleton structure?
Microtubules.
Very nice. Which is the most permanent?
Intermediate fibers.
The intermediate fibers. And then the microfilaments are found closest to the surface. What do the microfilaments do? They help the cell to change its shape. Does anyone happen to remember the protein that microfilaments are made of? Say it. Not, oh, you get a unicorn high five right there. Actin. Microtubules are made of tubulin and they are tube shaped. They're the largest of these three structures and we can disassemble and reassemble them as needed. These are what our organelles will anchor to. So they'll use them to kind of help guide them through around the cell. So like for example, the cell says, oh no, we need more energy in the southwest quadrant. Mitochondria, please help us. Mitochondria, they can't swim, they don't have legs, they don't have a tail. How do they get to the southwest quadrant? It's kind of like, think about an astronaut in space. If you've ever seen when they get stuck, like if, it was really a very funny video. So this astronaut was up in space and he got stuck in the middle of the room and he couldn't reach either of the walls. And he's just floating there. And what do? So, I mean, you can't, no matter how much you move, guess what? You're not going anywhere. That's terrifying. So that's kind of what our mitochondria and organelles are doing. They don't have the ability for motion. So they need these highways, these anchors to kind of pull them along just like a spaceman. The microtubules give them this ability and then they can anchor themselves and be in the part of the cell that they need to be in. The intermediate fibers, mama bear, these are the most permanent and these act, remember, like guy wires. They provide opposing force to help stabilize the shape of the cell. We had two different names here. These are both intermediate fibers. These two different names are only telling you what kind of cell are they in. If we call it a tonofilament, they're an intermediate fiber in an epithelial cell. If it's a neurofilament,
Nerves.
then it's in a nerve cell. We're still talking about the same type of fiber. We're just now discussing what kind of cell is it in. And then finally we had baby bear. The smallest and most delicate. They're woven like a spider web around the outside of the cell or closest to the cell membrane. They are made of a protein called actin. And they help the cell to change its shape. They can disassemble and then reassemble, and they play a key role in changing that cell's shape. Is everybody okay with the cytoskeleton? So then we started talking about the organs. The mitochondria, you know, creates energy. Creates energy in the form of ATP. Somebody remind me what ATP stands for. Adenosine triphosphate. That's where we have three molecules. We break the bond, release energy, reform the bond. Break the bond, release energy, reform the bond. Where this happens in the mitochondria is called the cristae. All that is the location where our energy is being produced And it's also where our cellular respiration is happening. Cells have to breathe just like us. They don't have lungs, but they do have to take in oxygen and expel carbon dioxide. And this is where that happens, is in this cristae. So again, we're just dealing with definitions. Mitochondria can replicate themselves, but they don't do it by mitosis, which is when a cell makes an exact copy. They do it by fission. And they just kind of bloop, pinch themselves, and then bloop, make two. So our higher energy cells that need more mitochondria are able to divide to meet that need. Here's the cristae. You see these little folds. These little folds help to increase the surface area inside. And that's where the ATP and cellular respiration. Ribosome is the little factory of the cell. His job is to make proteins. And so when our body says, hey, we're getting low on this particular protein, DNA starts, because remember, DNA is kind of the overseer of everything. He writes out the recipe and he gives it to someone. Who does he give it to? Well, at first he's got to get it to the ribosome. Who takes it there? RNA. Our RNA. RNA leaves the nucleus and goes and finds the appropriate ribosome. And he reads it and he starts assembling, kind of knitting or crocheting that protein together, and then releases the protein for delivery. They're the most numerous organelle in the cell. There are more ribosomes than anything else because their whole job is to create stuff that we need. The ribosomes primarily are found on the endoplasmic reticulum, specifically the rough endoplasmic reticulum. It's called the rough endoplasmic reticulum because it looks bumpy. Look at all these little knobs. That's where our ribosomes are created. The ribosomes can either stay stuck, stationary, or they can be like, I'm out of here, dude, and they can go just float around. So they can be free or they can be stationary. And this endoplasmic reticulum is just simply an extension of the nucleus. Now we also have the smooth endoplasmic reticulum. Tell me what we decided that was for.
Like it works like the kidneys detoxify.
It
Tolerance.
detoxifies. Yeah, it gives you tolerance. We compared it to having many beverages. And so when you have the burrs, it acts kind of like your kidneys. This is the kidney of the cell, detoxifying the cell for you. And if you have too many burrs over a long period of time, the smooth endoplasmic reticulum will actually get bigger. And that's what gives you your tolerance. You say, okay, now obviously our animals are not drinking alcohol. Hopefully. They should not be. But there are drugs that do the same thing. I think the one we specifically mentioned was like phenobarbital, which helps with seizures. And over time, the animal will increase the amount of this smooth endoplasmic reticulum, develop a tolerance to the drug, and suddenly you have to be giving more of that medication to achieve the same result. So there's just our rough ER. We've got our assembly of ribosomes. Smooth ER also helps to create and store fats. The Golgi, what did we compare the Golgi to? FedEx. So we've now got our ribosome has created a protein, now what?
Yeah, I gotta ship it out.
Now we gotta ship it wherever it needs to go. And so I put it in its little vehicle. This vehicle is called a vesicle, which is a void. And this little void will take it to the Golgi, and the Golgi will decide, okay, this has got to be shipped outside of the cell, or this has got to be shipped somewhere else within the cell. And he packages it up, puts his little stamp on it, and then loads it onto the appropriate truck, another vesicle, and it'll move it to wherever he stamped it to go. That's the whole point of the Golgi. He just repackages and designates where does this structure need to go. So here's a great picture. Here this is my ribosomes. They're catching a ride with their protein. Here's the vesicle, that's their vehicle. Drops them off here at the Golgi apparatus. They drop off the protein. Golgi says, okay, here's where this needs to go. He repackages it in a new transport vesicle. This particular little group of proteins was scheduled to be leaving the cell. So we take it, we bind this with the cell membrane, and we shoot them out into space. Lysosomes, these were the destruction. These have an enzyme that will destroy debris and foreign invaders, but at the same time also destroy the cell at the right time. As the cell ages, we get more and more of these, and when they are signaled, they all release their enzymes at the same time, and it causes destruction of the cell.
Breaks it down.
Which is what breaks it down, and tell me what the term is for that. Autolysis. Auto, self, lysis, breakdown. Sometimes this guy is also known, just in layman's terms, as the suicide cell. So we've got a little void, our vesicle, our inclusion, has found some nefarious creatures. Oh dear. Our lysosome comes on over, binds with it, releases his digestive enzymes, and breaks them down. Peroxisomes, peroxide, right? It's right there in the name what it does. It takes these nasty little free radicals. When do you get free radicals? All of the times. All of the times. When you have any kind of cell damage, you're forming free radicals. And when do you have cell damage?
All the time.
All of the time. All of the times you have cell damage. So you have these little free radicals going around trying to create more damage. And so the peroxisome catches them and converts them into what? Think of just the name of this.
Peroxide.
Peroxide, absolutely. But now, do we really want my cell filled with peroxide? No. So we're going to convert it into water. Excellent. It catalases. Remember cata?
Down.
Yeah.
Down
To-
under.
Down. See? It works. Down, under, against. Catalase means we're breaking it down further into water. Nice job, guys. And then inclusions are just a void. It's just a space. We can use this space to transport things. We can use this space as like a stomach to break stuff down. You have one cell where you can physically see the space, the monocyte, where it has these little voids, these little inclusions. And depending on the size, that'll determine the name. Sometimes we'll call them vacuoles, which are humongous voids. vesicles or very small voids, but it's just a space in the cell used to either trap invaders and debris or to transport stuff around the cell. Centrioles. This is going to be an important one when we get into mitosis. Because essentially what happens is these, I have my, when we're getting into mitosis, I have 2 exact copies of DNA. Here's 1 copy, here's 2 copies. This is called a chromosome. But I want one of these copies to go to one cell, and I want this other copy to go to a second cell because I'm replicating. How do I pull this apart? All right, I get my centrioles to line up, and they go pew, 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 and they form these little spindles. And then they tug of war, pull these 2 apart, and I end up with 2 identical cells. So that's what the centrioles do. And then finally our nucleus. This is where all of our DNA, the blueprint, the planning guide is kept.
Consider the yolk of the.
We looked at it like the yolk of the cell of the egg. Do all cells have a nucleus?
No.
No, they don't. Some cells don't, because for why? Because they're a prokaryote and they need to transport stuff. Those are 2 excellent answers. Prokaryotes don't have a nucleus, and our transporting cells don't need one because their job is just very passive. They just pick stuff up and deliver it. The nucleus would take up too much room, and I would then lose the space to transport stuff, and then I would have to have too many cells to do the same job. So exactly, that's exactly right. So this is just the anatomy of the nucleus. Here's our nucleus right here. We have the envelope, so the nucleus has its own little bit of skin. It really does kind of look like the yolk, doesn't it? The nucleoplasm, all right, so what did we say cytoplasm was?
Everything inside.
Everything inside.
Enzymes, proteins.
Absolute, so nucleoplasm,
It's gonna be the jelly inside the
everything
nucleus.
inside. The jelly, the proteins, everything inside the nucleus. The chromatin. All right, remember what I just said, chromosomes? What were chromosomes made of? DNA. Chromatin is
Hold the
just
DNA.
DNA. And then the nucleoli, which is a yolk within a yolk.
Which would be like the core inside.
It's kind of the core.
Blueprint. That's no, sorry. I got this written the wrong way.
That's okay, we're gonna go over exactly what these 2 structures are right now. So the envelope, which is the skin of the nucleus, has these little openings so our RNA can freely get out and deliver stuff to the factory, which was what? Who was the factory?
Gold apparatus.
That was the, okay, so that's the shipping. Who is the factory? The RNA comes down, and then he makes the protein. He's the most numerous organelle in the body. There you go. Ribosomes are the factory. Golgi is the shipping.
So, different type of factory.
Absolutely. So our nucleoplasm is going to be this jelly-like protein-filled substance. Chromatin, these are just our DNA, think chromosome, think of those connections. And then when we are dividing our cell, We super hypercoil them and turn them into a chromosome. So that's just chromosomes are chromatins, super, super concentrated, ready for division. We're going to talk a lot more about that in chapter 4. And then finally, we have the nucleoli. And this is what contains a very specific DNA that creates the RNA. So our RNA, our ribosomal RNA, the one that goes out and seeks out a factory, well, we got to have a place to make that. And this nucleolus is where that happens. Makes ribosomal RNA, which is who takes the recipe out to the ribosome, lets the ribosome read, creates the protein. No, the DNA is already there. And that is that. All right, I've got about 10 minutes left. Here's what I want to do. I'm going to turn this off. We'll test questions. Give you an idea of what you might expect. They may or may not be actual questions on the test.